So today I'm going to talk to you about a method that we started developing to demonstrate epitaxy from supraatomic resolution images. So what is epitaxy? Epitaxy is the oriented growth of an overlayer, and it is due to atomic scale interactions with the substrate. And it's important in the study of structure microscopic property relationships. And so our question today that we want to address is, can we detect epitaxy without atomic scale information? So first of all, there are actually different sources of oriented growth. So for example, there can be local self-templating, and in that case we'll have regions of different orientations of our crystallites. We can also have preferred orientations due to some asymmetry in the growth process, for example, the flux of the deposited material. And finally, we have our epitaxial oriented growth due to epitaxy, where the direction of the growth of our crystallites is dependent on the substrate orientation. So in order to discriminate between those different sources of anisotropy, so we thought about this little sort of decision tree or algorithm for experiments. So once we observe preferred orientations on a surface, we first test sample homogeneity to see if we have local self-templating. And after that, we can test sample rotation during the growth of the overlayer. And if the growth doesn't depend on the orientation of our substrate, we can say it's probably epitaxy. Now, in order to do this, we need to first observe preferred orientations. So we're going to do this in an automated way. And so we start with our data, which is digital image. And then we'll detect the step edges, which then we will associate a local edge angle. And from that, we can compile all the values and get an angle distribution, which we can fit to identify preferred orientations. And finally, if we know the orientation of the substrate, we can refer that angle directly to the crystal, the substrate crystal. Okay, so so how do we do this? So I would say the most common technique to identify edges is to use the derivative, the local derivative. So our edge is actually signaled by a peak. Now let's apply. So how do we do this more precisely, or specifically in my test case? So I use the what's called the Sobel kernels here, that allow us to calculate the local gradients in the x direction, so in the y direction, by simply convoluting this matrix with a local 3x3 matrix of the image. So those are the pixels. And so this looks very much like a numerical derivative. And then what we do essentially is that we scan this operation all over the image, and then we can calculate the local gradient and also the edge, the angle of that gradient. And we get a spatial distribution of both those quantities. Okay, so our test system today is this holmium oxide film on sapphire. And so this first image has about 100 features to look at. So we apply our gradient filter. And so this is the image, and this is the normalized gradient, so values from 0 to 1. Now, if we look at what those values represent, the lower gradients, so the smaller slopes, are essentially the facets, right? When the higher gradients are showing the edges which we're interested in. So what I did is to really sample the smallest portion of this gradient that shows the thinnest possible edge without losing too much signal. And now from that, we can compute the angle distribution and collect it into a graph. And so what we see here, we are able to fit a certain number of peaks that seem to indicate that there are preferred orientations for those edges. Now from that, we can also plot it back on the image, which here shows all the crystallites with the different edge orientations. Okay, so it looks like on this image we have 3 families of preferred orientations. So now we talked about sample homogeneity earlier. So this is the next test that I've performed. So the previous image is this square in the corner. Now we extend the analysis to this larger image, which contains about 1,000 features. Now, we simply split the image into the same size images as our original one. And if we plot the compiled edge orientation distribution, we get something like this. So clearly there seems to be a trend here across the whole surface. But can we look at it a little bit more quantitatively? So to do that, I used cross correlation between the different angle distributions. So you have an image A and an image B, and each of them have a certain distribution. And so the cross correlation is simply like scanning one of the distribution over the other and finding the best match. So I think the advantage of that is that it's not sensitive to added background or dilatation or like multiplication factors. So this here is the result for cross correlation of that first image I showed you and all the other ones. And so what we can see first is that there doesn't seem to be a loss of correlation that is correlated with the distance. So it seems like it's pretty homogeneous. Now we can also look at all the other pairs of images. And this is the result. So each square is, so this image and its cross correlation with all the rest, etc. And I think this particular representation allows us to visualize if there are outliers there. So for example, this square of less correlated image is reproduced here. So this subpart of the image is an outlier. But generally, we have pretty good cross correlations. And if we plot all the pairwise cross correlations, we find that we're hitting towards 0.8 correlation, where 1 would be the perfect match. So it looks like for this sample, we have preferred orientations that are pretty much homogeneous, at least over this area. Now, so this is the beginning of this development, this method development. So we, so far we have an automated edge orientation detection, and we can detect spatial homogeneity pretty fast. And our next step will be to test process anisotropy. So that involves another deposition experiment in different orientations. And then also look for broad applicability, test different substrates and possibly other types of data. So far we looked at an AFM image, but why not SEM or optical microscopy? And this is the end of my talk, and I'd like to thank you for your attention. And if you have any questions, I'll be happy to answer them.
Open for questions.
Okay. How are you getting the angle out of the image? Because I saw there were a few almost just single spots that seemed to have an angle allocated to them. I could see the longer edges that something might fit through, but like there were just specks that didn't seem to have any orientation.
Yeah. So you're talking about this?
Yeah, I just see like tiny single red points and so on.
So in fact, this is both the map of the angles filtered through my my gradient pick. So that's why. There are data points for the angle all over.
But how does one decide what edge angle it is when it's one isolated point or a couple of isolated points? I would have thought it would be hard to know what the angle is of a single point, or am I?
Yeah, I mean, there is some noise, definitely. I mean, so the testing, so I tested the method also on a synthetic image of triangles, and we still get a spread in the preferred orientation. So there's some error in there.
What was the gradient acceptance kind of criteria that you used?
Yeah, so it's simply a cutoff. So 0.252.4? Yeah, so it's with the normalized gradient. So everything is divided by the highest value. And then I pick a certain slice. This is done visually mostly. I look at how does it look. And if it describes the edges and it's thin enough in an effort to reduce errors.
Something that needs work on, like automating that, or have arbitrary objective ways to choose that criteria. Perhaps most likely based on the iterative process of comparing post-selection, cross-correlation, and
always choosing those kind of numbers are important.
May I make a comment?
Yep.
I think it's very nice. I understand your purposes without doing, all this other experimental work, just, using your methodology to find out preferred orientation. But I think the edge may not represent the real crystallography orientation, because there's a whole community talking about epitaxy and what's a preferred orientation. The most straightforward way they did is using the X-ray diffraction map out azimuthal rotation. That will give you precisely this tree orientation that you are talking about. I would suggest you check literature, because you made a comment you want to use optical way, other math or AFM, but I think diffraction may be a little bit easier for you to compare with your methodology.
See, as a test.
Because I think it's very nice if you have this methodology. It will be even nicer if you can prove from, first of all, is it crystalline? Because you need to have crystalline, you want to do X-ray diffraction. Is it crystal?
Yes.
Is it crystal?
Yes.
Okay. And you can just do a simple azimuthal rotation of your, sample. You confirmation of orientation distribution.
do you guys have such an instrument?
Huh?
Do you have such an instrument? Perhaps we could.
Or if you ask your colleague to do X-ray diffraction, they all know.
Okay.